Good evening, everyone. Ladies and gentlemen, welcome to Motor Health, Transatlantic Cooperation Strategies and Tech Transfer in Sensorimotor Neuroscience. I am Eva Bosbach, Executive Director of the North America Office of the University of Cologne. And it is my great pleasure to open tonight's event, where we will have the opportunity to learn about the fascinating work of 4 scientists on the panel, as well as about strategies for transatlantic cooperation and tech transfer. Around the world, millions of people live with motor dysfunctions caused by neurological conditions such as stroke, Parkinson's disease, or spinal cord injury, impairments that severely limit their independence. The science behind motor health is extraordinarily complex, positioned at the intersection of neuroscience, engineering, and clinical research. And the goals are also very ambitious and noble: to restore movement, to reduce long-term long-term care burdens, and improve the overall quality of life. Today's panel focuses on how we can advance those goals through international collaboration and enhanced dialogue between academia and industry. As a university committed to global partnership and interdisciplinary research, which you can see on our 5 clusters of excellence and also 5 international liaison offices, so except for the one, in addition to the one here in North America, we also have one in China and one in India and 2 in Africa. The University of Cologne is proud to host the conversation with colleagues from the US tonight. Transatlantic cooperation is not just an academic ideal. It is a necessity if we are to accelerate innovation, scale new technologies, and ensure that they reach the patients who need them most on both sides of the Atlantic. This event would not be possible without our wonderful partners, the German Center for Research and Innovation, DWIH New York, the German Research Foundation, DFG North America, the German Consulate General New York, and our host here in the building, NYU Langone Health. So thank you all so much for your generous support. Tonight's event was initiated actually by 2 researchers with shared interest in motor health, whom will be here in just a few minutes, Dr. Graziana Gatto from the University of Cologne and Dr. Alice Mosberger from NYU Langone Health. And I think we can say that their collaboration is really a shining example of how transatlantic exchange can bring together complementary expertise, pool resources, and open up new research frontiers. We are honored that we can be here tonight. And as a final note, I would like to thank everyone who helped organize this event. Thank you, Alice, Heather, it's probably still happening downstairs, Jessica, Cara, and Julia and Saskia also from the DWIH team. So it is now my pleasure to give the floor to Dr. Jan Lüdert, Head of Programs at the German Center for Research and Innovation, DWIH New York, for his welcoming remarks. Thank you, Jan, and everyone for being with us tonight.
Okay, I think this is the spot where I need to stand. Good evening, everyone. It's a pleasure to welcome you all to tonight's panel discussion, Motor Health, Transatlantic Cooperation Strategies and Tech Transfer in Sensorimotor Neuroscience. On behalf of the German Center for Research and Innovation New York, DWIH, I want to express how proud we are to support this event, organized together with our partners from the University of Cologne, NYU Langone, and the German Research Foundation North America, as well as the Consul General of the Federal Republic of Germany here in New York. So if you're interested, there's a Schnitzel Pass. You can come to our restaurant and enjoy great schnitzel at our house. I think, Nina, you've been there several times?
Several times. Monday, Wednesday, Friday.
We don't bribe anybody. We'll have you come for schnitzel. At DWIH New York, our mission is to strengthen German-US collaboration in research, innovation, and entrepreneurship. I think all of you in this room know how important this is, especially in times like these. We connect universities, research institutions, startups, and policymakers to advance scientific exchange and foster solutions to global challenges. Each year, our programming follows an overarching annual theme, and this year's theme is global health. Would not be a more relevant topic than what we are about to hear from our esteemed panel tonight. Motor health and sensory motor neuroscience stand at the intersection of healthcare, technology, and international collaboration. Across the world, millions face motor dysfunction stemming from conditions such as Parkinson's disease, stroke, or spinal injuries. Understanding and addressing these challenges requires not only groundbreaking science, but also spirit of cooperation, precisely what we aim to highlight this evening. Our distinguished panelists embody that spirit. They represent laboratories and institutions spanning Cologne in Germany, with an amazing cathedral, which you have to see, New York, with also a lot of amazing things, Atlanta and beyond, where I will be going next week for the first time in my life. So if you can tell me anything about Atlanta. Great. Working across disciplines from neurobiology, engineering to computational modeling and biomechanics. Together, their work raises a lot of vital questions, which our esteemed moderator, Nina Gray, will probe them on. But these are not just scientific questions which we're going to embark on today. They are questions about how we organize knowledge, fund collaboration, and share discoveries across borders. I'm confident that tonight's discussions will illuminate all of us. And with that, please join me in welcoming our moderator, Nina Gray, Associate Vice Provost of Research at New York University, who will guide us through this exciting evening. Thank you so much.
So we'll get to Nina in just a second. I just want to point out this is the cathedral that he mentioned, and you can come for the schnitzel and also visit our office. And also, if anybody is thinking of research collaborations with Germany, student exchange, please talk to me after the reception. Also, we have handouts outside about research in Cologne and about funding opportunities. So please take advantage of that later as well. Before we start with the panel, we have one more highlight for you. We actually have Dr. Georg Bechtold from the German Research Foundation, North America, who has been engaged in the promotion of European and transatlantic research cooperation since he joined the DFK in 2003. And we are very happy that he can share his knowledge about transatlantic cooperation and funding opportunities with us in a short presentation tonight. So while unfortunately Georg could not join us here in New York today, he's live with us on Zoom. So welcome, Georg, and he will give us a little keynote, and then we'll get to the panel. I will introduce Nina right after that. Georg, can you hear us?
Yes, I can. Can you hear me?
Yes,
Perfect.
you can go ahead.
Good. Yeah, thank you very much, Eva. This will be a kind of advertisement block, and please stop to talk about schnitzels because I'm really hungry. But it will take just 7 minutes, so I will survive it. Right. Good evening, ladies and gentlemen. My name is Georg Bechtold. I'm from the German Research Foundation. The German Research Foundation is comparable to the National Science Foundation, which you all know from the United States. But we don't fund only STEM research, but also research in humanities, and which might be of relevance for you, medical research. Today, this evening, I would like to introduce you to 2 programs where people from the US are under certain circumstances are eligible to apply at DFG. This will be the Walter Benjamin program for younger postdocs and also the Emmy Noether program for more experienced ones. And on top of that, we'll talk a little bit about the individual grants program where US citizens are not allowable, but German citizens, and they have some ways to include their US counterparts. So let's talk about Walter Benjamin. The Walter Benjamin program is meant to fund researchers that are about to finalize their PhD or just have finished their PhD. And it's possible for US citizens to apply at DFG if they have a host in Germany for a research stay of up to 2 years at that host in Germany. And good thing is it's not just a fellowship, but it's a real position. That means you get a real salary, of course, together with social security and also health insurance and all that things. So a real salary in Germany and also a small allowance for direct project costs. Yeah, the only thing you have to do is to find a host in Germany, and that host institution needs to provide all additional project costs, of course, in the case if you have been successful with your proposal at DFG. The second program, the Minuta program, is for independent junior research groups. So if you have finalized your PhD the last 2 to 4 years, and most of you might be in medical research and in psychology, even up to 6 years after your PhD, you can apply for that junior research group. However, you need to have a substantial international experience, for example, maybe some stays abroad of the United States. And of course, you need to apply for an excellent research project, and the chances for success are around 20%. So once you're successful, you get funding for up to 6 years with your own position, again with health insurance, social security, and also all that things with a quite good salary. And on top of that, you can also apply for staff, for example, for a postdoc, for a PhD student, or if necessary, for 2 or 3 of them, for consumables, smaller instrumentation, but also for a travel budget to go back to the United States from time to time, to keep your networks, and also for guest visits to invite people from the United States to visit you at your research group in Germany. So the third program, as I said, it's our bread and butter business. It's the individual research grants program. And there, any scientist can apply, any German located scientist can apply at DFG for a project of up to with a duration of up to 3 years. And funded is staff. Typically, it's a PhD student or a postdoc. Consumables, and as I already mentioned before, travel costs to travel to the United States, guest visits to enable visitors to visit them in Germany. And also very interesting, I guess for you, is the Mercator Fellow. That means you could become such a Mercator Fellow if your German partner is successful within such an individual research grant program, and they can apply to invite you to come to Germany for a period between 3 and 12 months, paid by DFG. So if you think about a sabbatical, that might be the right solution for you. And on the next slide, this is the perspective of your German partner. If you have a joint idea for a research project, for a joint research project, I will show you this flow chart here. I like that. The first question you have to ask, or your German partner has to ask, is the share of the North American partner low? And if that's the case, for example, if it's just an analytical method or something like that, then your German partner can apply at DFG, for example, within that individual research grants program, can apply for additional travel grants to visit you in the United States, and also the Mercator module I just mentioned to enable you to have a fellowship in Germany here. If that's not the case, if the US partner needs considerable funding, the question is, could be the German part and the US part, could they be managed somehow that they are both independent on each other? And if that's the case, the solution is easy. The German applicant applies at DFG, and the North American applicant applies, for example, at NSF, or in your case, maybe at NIH. And if both of them are successful, it's good. If only one of them is successful, it's not that good, but at least this person can then run the project at his part or their part. Yeah, but of course, it's much better if the project is completely, if the US part and the German part are completely intermingled somehow. And then there might be a chance that the US partner has their funding already available, for example, by their own university or so, or other sources. And if that's the case, you just need, just send the confirmation to your German partner, and they apply for DFG at DFG. And if they're successful, you can run your project. Unfortunately, the flowchart ends here. And if that's not the case, unfortunately, we don't have any joint programs with the NIH. So you have to start again and have to get another idea. Anyway, that's what I can tell you, and I hope you are interested now in that 3 programs, and maybe one or the other has some questions, and I stay here to answer them. Thank you very much. I'm Georg Bechtold. Thank you very much.
Thank you so much, Georg. So this was actually very enlightening and also seriously meant. And
Thank you,
I'm
Eva.
sure people can also scan the QR code and contact you later on if they would like to get more information or maybe pursue one of those pathways. Thank you so much for showing us how it can be done. So I would now like to hand over the mic to the moderator of today's discussion, Dr. Nina Gray, Associate Vice Provost for Research at New York University. Nina works with leaders across NYU's 3 campuses and 17 schools and colleges to foster interdisciplinary research, knowledge creation, scholarship, and innovation through partnerships and collaborations, and by prioritizing integrity and inclusion. Nina, you and I have done a number of events together, and I'm very thankful for our friendship and also for the fact that you are also a strong promoter of transatlantic collaborations and devoting your time to events like these, but also to other formats such as the DWIH Future Forum or the Falling Walls event. So thank you for that, and thank you for being our moderator tonight.
Thank you very much. And I'm happy to get things started on the science front. I'm going to ask each of our panelists to introduce themselves briefly and tell us a little bit about the work that they do. Then we're going to dig into a little bit more details of that work later. But I think we were going to start with Anna, because you have a microphone in your hand. And this was your idea.
So this was my idea because we were discussing like, what's my science? How can I promote it? And so first let's get into the science and then why I chose these particular people and why collaboration and an integrative approach are very important. So I've always been fascinated because we move. Everybody can move. We do a lot of complicated stuff. If you're an athlete, you can jump around. Right now, even if I'm still, I'm actually moving because my body has to keep an upright position. So I have to hold the microphone and I have to speak, and I'm doing all these complicated things at the same time. And this is because there are neurons, so little cells in our brain that work together to make sure we do this correctly. And we understand how this is important when things start not to function. For example, it was mentioned Parkinson disease or spinal cord injury, and we cannot move anymore. So what we do in my lab is really trying to understand how we do all these things to understand how we can repair them when these things go wrong. And in particular, we work not on the brain, but in the spinal cord, which is here inside your vertebra, is a long tube full of neurons, but they ensure very important thing. They ensure we breathe, we ensure we move our hand. I'm Italian, so this is gonna move a lot. We get special circuits for that. But then they also assure that we can understand if we touch something, if this is like a good hold, or for example, if now we walk out and it's gonna start to rain, the road is gonna become very slippery. And then we know we have to walk differently. And you know, it's also good because sometimes we want a hug and then somebody's gonna hug us and that's gonna feel very good. But it's also good for warning us or animals in general about danger. So I think everyone is familiar. If you wake up, you're still a bit sleepy, there is coffee coming out of your coffee machine, and you touch it, and it's hot, you know that the best thing to do is to just... And the reason is you don't even think about it, because in the spinal cord, there is this circuit that is like protective and will tell your hand exactly what to do, even if you don't think about it. And so what we do in the lab is really understand how we feel cold, we feel touch, how we feel where our hand is in space. That's called proprioception. It's our sixth sense. And also how all this sensation makes our action so perfect, so that we can walk, talk, hand move. And this is also like, we do it continuously in our daily life. And if we understand how we do all these things, we can make sure everyone can do it. So we can promote motor health. And before handing over to the next speaker, I wanted to touch upon on why it's important to have this collaboration. And the thing is that the brain is a very complex machinery where a lot of neurons work together to make it work. And science also requires a lot of different expertise. So one of the reasons I met Victor, I will also explain why I picked these people on the panel. So when I was doing my postdoc, I met Victoria, and we were she was at Harvard, I was at the Salk Institute in San Diego, and our lab decided to have a joint 2 days retreat. And Victoria is actually the expert on sensation. She can tell you everything about how you feel touch, and I was the expert on how we move. And we thought, okay, maybe we should work together, because then I understand better touch, you understand the better movement. And this was in 2016, 17. And then now we have our own lab and we keep working together. She visited me in Cologne, was very transformative because we came up with a lot of idea and project we can do together. And so then she will tell you more about her expertise. And also while I was a postdoc, I met Sam, which was just came for a visit and a talk at Salk. And he was having this new electrode that we'll talk about. And then I just asked him a question, and I think I was supposed to talk to him for 5 minutes. We ended up chatting for 2 hours. I completely destroyed the schedule. And then we have kept working together because the way our lab are very complementary. We have a lot of ongoing projects. I think we meet on Zoom every other week. And then while I was designing this electrode with Sam, he said, well, there is another person that's using them, Alice. And we started to have this Zoom where we discuss like, okay, this is how we do it. How do you do it? And then sometimes we had a good idea. Most of the time, Alice had better idea. But we really, it's like how this collaboration work. And I think it has made all our science better, but also it's a lot of fun, right? Because then you get to hang out in New York when you don't think you would have otherwise.
Great. And we'll pass it to your left.
To Victoria.
Yeah.
We go to neurology.
That was actually
like
my job. I'm like, hi, I'm Victoria. She summarized it. So I'm Victoria Abraira. I'm an associate professor at Rutgers University in New Jersey, which is actually like the Garden State to New York. So we're kind of like all one big family. And so as Graziana mentioned, my lab works on the sense of touch, really. So the sense of touch, what I find fascinating about it is the first sense to develop and the last one to die. It dies with you, right? So it's one of the senses that is with you, for the entirety of your life. And it wasn't really until I started, working with Graziana and collaborating with folks that I realized that, the sense of touch is really important for movement, right? So this is where then my lab started to become interested in, how do we, how does touch help us move? And that really sort of generated really aspect of, research in our lab, but we now work on spinal cord injury and recovery from injury. And so I think for me, these types of collaborations end up opening new avenues for our research, but also it allows you to take a research area that you're really interested in and then asking questions like, well, how does it interface with these other areas of research? And as Graziana mentioned, the brain is really complex, right? So then it takes people from various different, training backgrounds to really come together and really figure this out. And so I'm really happy that we met, and started this 10 years ago. And here's to another 30, 40 years. I don't know how long we'll be alive, but hopefully for a long time. Okay.
All right, thank you so much. Yeah, so I am an assistant professor here at NYU Langone. My name is Alice Mosberger. I started my lab only just recently. And I also do research in my lab that involves what we call sensorimotor control. So how does the brain integrate this sensory information and produce movements? And my lab is particularly interested in understanding how we produce skilled movements with our arms. For example, when you reach to your phone on the nightstand in the morning, that's like one of a very well practiced, potentially skilled movement. And we're particularly interested in understanding how the brain can actually do that using different strategies. So in motor control, we talk about these feet forward or feet back type movements. And the idea really is that a feet forward type movement is a movement that is really well practiced and fast. So you can imagine Roger Federer swinging his tennis racket to serve the tennis ball. And it happens so fast that the brain doesn't actually require that much sensory information to do it. It's like automated to some extent. And then there's other movements that are more feedback based, and they are more continuously requiring one of these senses in particular that Graziana talked about, that is proprioception. So the sense that your brain has of where your limb is in space. And to produce movements like drawing a smooth line on a canvas, for example, you need that sense. And this is a feedback-based movement, we say. So there's these two extremes of these movements, but even a single action or any movement you do sort of like exists on the gradient. So the brain can actually do it using one strategy more or the other strategy more, depending on the sensory information that's available. And I'm particularly interested about this because it also allows the brain to have what we call degeneracy. So actually the brain is so complex and it has so many different circuits that it can produce the same movement using different subcircuits. And that allows the brain to also be resilient against injury. So if you have a spinal cord injury or a stroke, parts of your brain are not working anymore. You lose some function, but you can then recover that function by retraining the other circuits to overcome this and learn to produce a movement in a new way. And so my lab is really interested in understanding that. Yeah, and I just wanted to mention also, like, why do I think these collaborations and multidisciplinary research is so important? It's really because we have sort of become like what we call systems neuroscientists in the past couple of decades. And so we try to understand the organism as it behaves and the cells that are important and the proteins and the genes, sort of the whole gamut of biology. And this requires really multiple expertises. So we require engineers that understand the math behind movement, and we require biologists that understand how do we look at the cells, and geneticists that can then inform us, oh, this cell maybe has a specific gene that causes a disease, and to work across these different disciplines and across different nationalities as well is really what enriches our field.
Thank you very much. So my name is Sam Sober. I'm a professor of biology at Emory University in Atlanta, Georgia. I am sorry to say that Atlanta does not have a cathedral to rival Cologne, but what we do have is a lot of incredibly good barbecue restaurants. And I would argue that if the barbecue restaurants in Atlanta were a cathedral, it would look very much like the one in Cologne. So my lab is interested in how the brain learns new skilled behaviors and how the brain then executes these very complex skills. So one thing we study is how songbirds learn how to sing. So we study how the neurons in a songbird's brain communicate with the vocal muscles as a bird is first learning. We also work on skilled limb control in rodents with a lot of common interest with people here. My lab also develops new technologies for examining how the brain can communicate with muscles, both in healthy individuals and also in states of neurological disease. So these technologies that we've been developing, actually in collaboration with people up here, as Graziana was saying, So these are things that started as a basic research tool, and we've expanded. We've provided these devices to hundreds of labs around the world. We've run training courses, including at the University of Cologne, to build the community of people who are using these devices. And then we're also translating these devices currently to be clinical tools. So it's been this great, international experience of creating new devices and new technologies that can help push neuroscience forward, both for these, curiosity-driven things like, how does a bird learn how to sing, and also for clinical applications. So, this is driven by the intuition that it's the same brain processes that allow a songbird to learn how to sing, that helps a young child learn how to speak, that helps someone who's recovering from a stroke or a spinal cord injury to learn how to move their body. So thank you very much, and it's nice to be here.
Fantastic. I have a gazillion questions, as I'm sure the rest of you do too. I'm going to try to stick to my notes and not go off the reservation. So all of you talked a little bit about basic research and the basic understanding of neurons, connections, and how that works in sensation, proprioception, movement. And you also talked about disease and disorder. And so I want to start by getting into that progress from doing basic research to translational research to maybe not quite bedside, but clinical trial, and what that experience has been like for you. I think most of you maybe started as basic researchers and have gotten into the translational. So what did you have to learn, and what are some of the challenges that you faced moving into that more applied space of research?
Well, actually, my experience was actually the other way around. It's kind of interesting because I did my PhD in a more translational field. I worked in a lab that exclusively studied spinal cord injury and then moved into stroke, doing basic research, but also really with a very strong connection to the clinic. So one of the things that I found that really rewarding in my PhD was that I had regular interactions through foundations, but also just through collaborations. This was in Zurich, Switzerland, actually, with clinicians and with patients. And I think that is something that actually excited me a lot coming here to NYU Langone for starting my own lab, because we are at the medical school here and the clinical departments are very close and the patients are here as well in this building. And to have the interaction as a young scientist to really learn what do the patients actually care about, what are they struggling with, what is the experience of not being able to move or losing a function of your brain like, and what are the hurdles to overcome has been really informative for my research. And so I think that's something that we should foster across or with our trainees as well, to get this experience.
Yeah, maybe I'll just add, because I did actually the opposite. So I started very basic science. I was just working with cell in a dish and just looking what happens to them when we express a protein or they lack a protein. And then now in my lab, I'm actually at the University of Cologne in the neurology department, where all my colleagues are neurologists. So they focus on very clinical research. They have patients, we analyze patients. And for me, it was kind of transformative, because then I got to see, okay, this is what I do in the lab, but actually this could be a good application. How can I make people's life better? And sometimes, like, I always thought people with a stroke or Parkinson have mostly a motor problem. And then you talk with the patient, actually, it's more than that. They can't feel sensation as well in some cases. And sometimes they have chronic pain. And so I think it's broadened a little bit my understanding on how we do basic research. And also interacting with a different expertise, which is the clinical side, has been also quite transformative. And because as a scientist, we always think in our field, we ask our question, but then when you interact with somebody that thinks about the problem in a different manner, your way of thinking about the problem gets so much more deeper and multimodal and multidimensional, and makes you a better scientist and makes you do better science. So talking, for example, with colleagues, like when we do research, we start to get to that tiny little protein that you think if you change, it will have a transformative effect. Mm-hmm. And then you actually discuss with neurologists and they tell you which kind of drugs they do in the clinic. Mm-hmm. And they are not so very time specific for that tiny little protein you actually care about. And then you start to rethink the scale at which you do science. And I think for me, that's been quite transformative in the last years to interact with them. And then just to make this a bit lighter, also the other thing I've learned that patients wear shoes, mice don't. So when I do all my analysis, that's a big confound because there is a lot of variety of shoes. I think if you all look, some people have high heels, some people have flat shoes. And when we do our analysis where we have just mice walking and we just track their body, that's not a problem. If now you have shoes, that's a big problem. So I think it's nice because one, you learn new problems, but also I think it's very enriching as an experience.
Right. Yeah, sure. Go ahead.
Yeah, and I'll just add, I mean, so in the course of developing this technology that we created for songbirds and are now using in humans, this was really accelerated by international collaboration. So this project really took off at the beginning of the pandemic when it was incredibly hard to train people. I mean, the way that new technology usually spreads in neuroscience is, one lab develops it and then they share it with one or two close collaborators, and then, people sort of hold it very closely until the first papers come out. We really took the opposite approach of, we were fortunate to get some funding to provide these devices for free to these labs all around the world. And, we had, dozens and dozens of Zoom calls teaching people how to use these. And, remember, we invented these things to record from these tiny little muscles in songbirds. And we, at the time, we spent 10 years developing these things, never thinking beyond the songbird. But then we started sharing these devices with people working on mice and monkeys and lots of other kinds of animals. And we got tons of feedback from the international, our international collaborators for how to make the devices better. So it was, it worked out great. I didn't plan it this way, but because we were doing all of this international collaboration, it actually led to the technology getting much better, much faster than it ever would have if we had sort of pursued a traditional model of basically not sharing it until we thought it was as good as we were going to get it.
That's incredible.
Just one small thing that I think for me, just this past year, we were to do some work with humans and test some of our animal sort of models and animals in humans. What really helped is really, identifying a collaborator who ended up actually being in Sweden, right? So an international club, and then actually sending one of our trainees there. And I think people on this panel mentioned how critical it is for our trainees to get that training and to experience these, different training, different labs, and to really learn the language. And I think we want to approach translation, and that really comes with properly training the people in our labs to really think, at a bigger scale. And I think that does come with trainees, right? Going to these different labs and learning these things. So, yeah.
Right. That's great. We talked a lot about also sort of learning how it works, observing the behavior of the nervous system in different circumstances, but we're also talking about control and controlling the nervous system, and that being one of the sort of therapeutic pathways that is maybe most interesting or for some of us, right? So I'm wondering if this is a little bit off the notes, if anyone would like to sort of volunteer what they think we're on the cusp of as it comes to sensorimotor control. It doesn't have to be going into humans, but what's really coming out now that wasn't possible just a few years ago because of some of the kind of technological advances and the knowledge advances that you've seen?
The first, and then we can continue. So I think one thing that we really have learned to do is we generated these, what we call genetic tools. So it's just a way for us to make neurons activatable. So you can turn the neuron on or you can turn the neuron off. And that's a great function. So imagine you have a radio and now you can remove a part and then you see how the radio changes. And if you hear no sound, you know exactly what you caused. So in mice, what we can do is activate a neuron and then we see what happens. And for example, in my lab, we found some neurons that are in some part of the next to the spinal cord. We call them dorsal root ganglia. When you activate them, the mouse just starts to scratch. And so it will be the same as if a mosquito bites you. So now, because we know which neuron are the responsible for the scratching, we can imagine in the long term, okay, what's special about this neuron? Can we design a way to target this neuron for treating chronic itch, which, for example, is a side effect of diabetes? And so I think this is where we are on the cusp of, because we are able now to test which neuron does which function and then use this knowledge to see which could be a useful therapy. And the other thing we also know very well is that we can record, we can know when these neurons are active, because we can do, like, we can look at them in the brain and see when, you know, they fire action potential, or we can see how they are active during different things. So you can have a mouse walking and you see some neurons are active, and then you see these neurons don't care about if the mouse is reaching the microphone. And so this helps us also, depending on which disease, which neurons are better to activate to recover from that disease.
I might not add too much about like technological advancements in general. I think they're like, you know, we're recording from more neurons and we were able to manipulate them in huge scales compared to even just a few decades ago. And I think what's really exciting about the future is that we have also developed as a field more and more technologies to record that data and hold on to it and make it understandable to people in the field and across fields, to maybe clinicians or trainees at different levels, and to make it open source and share it early on is something where I'm really excited about that. I think we're doing more and more type of experiments where you have your question and that you have your animal do a certain thing, but you record from lots of cells in the brain, and maybe somebody else has other questions. And if they could access that same data, they would not have to repeat the experiment, but they could just look at that and analyze it in their own way. And with sharing these large data sets across institutions, and maybe there's some interesting discussion about how we could make that feasible more internationally and sharing data before publication and so on. I think that's really a great opportunity for trainees as well, because if you have a question, you can go to some of these data sets that have been shared publicly already and maybe answer it without having to do the experiment even.
Yeah, I'll add to that, just our ability now to analyze data, right? And so in the things that we use, so AI, right? Like it's in everyone's mind. So like, how do we utilize that to look at our vast amounts of data and make sense of it? And I think that's what's really exciting about translation, is because now we can do things and we can use AI to look at patterns in the data and things that we just weren't capable of doing before. So really look at, you know, previously, for example, FDA approved drugs for the treatment of X, but would it treat Y or Z, right? Like, can we use, you know, so I think that's super exciting, right? So, and so I think the questions there are like, how do we share this data, right? And how do we, you know, make it really serve the public, which I think that's international collaborations can do that, can define that for us, I think.
Turned into an advertisement for open science as well. Hashtag open science when we post about it for sure.
And yes, also it's been greatly facilitated by preprint. So open access journal where you don't have to pay a fee and you can just read about everyone else's research. Yeah, let's do the full advertisement.
Fantastic. Let's continue advertising about transatlantic collaboration next. And you all mentioned it, how important it is to be able to partner, to be able to share. I'm curious what you all think and we learned a little bit earlier about some of the mechanisms that help facilitate interaction between US and German institutions, but what kinds of mechanisms would you like to see from the US, from Germany, from foundations and other potential, you know, funders that could help promote this work and facilitate it to a greater extent than you've already been able to achieve?
Yeah, so one thing we were just discussing now, who says this? But one of the things we did is organizing shared training courses. So we had a training courses in Cologne where people from Europe could access and learn to use STEM technology. And that helped to diffuse it in Europe. We were lucky to fund this event because we had local funding. And, you know, but this should be a more consistent event. We want to do it not only in Europe, but across the world. And I think having a structure to fund this workshop, which are not only just to develop science, but also to train people, to make people network, and to get them also exposed to interact with scientists, that will be like the first thing of support, will be really great. But also to support consortia of scientists, which are international. I think there are few foundations that do that, but I think having more of these like intercontinental network will really help.
Yeah, and I'll just add to that. So this training workshop that we did at the University of Cologne, it was great for all the reasons that Graziana mentioned, but it was also fantastic for developing new I have a couple of new collaborations that started there and that have continued. So I think there's sort of the immediate benefit of you teach the people who are in the room how to use the thing, whatever the thing is. But it's really, the power of in-person training is that you develop these professional relationships that then, that then can lead to more things. And yeah, I mean, so, I've worked with, and that's been true, in that work in Germany. I'm one of the directors of this collaborative program in Canada that builds, US-Canadian networks. And, they happen to be focused on this technology, but it's really these in-person meetings that, wind up having these effects that sort of continue into the future.
Sabbaticals, right? So I did a month-long sabbatical in Graziana's lab, and that was transformational for my research program to understand, how does touch affect movement? So I think the sabbaticals, which you guys are already doing, which is so key. So, finding ways for people to connect, and even just some of these seed funding, often people think, is X amount of money enough? Even if it's a small amount of money, it can go a really long way, right? And what you really need to do is just get the scientists together, right? I think that's the main thing. So yeah, these sabbaticals for faculty and postdocs and students, I think is important. Yeah.
As somebody who gives out seed grants, I'm so glad to hear you say even a little bit helps.
No, even a little helps. Yeah, for sure. Yeah.
That's great. That's great. I'd love to open it up to the audience for questions at this point. As I said before, I have tons, but this is really your opportunity to ask more about the science, about the partnerships. We have one here in the middle.
I always have.
I'm Julia Kaiser. I'm a scientist also here in the New York area, and I'm originally from Germany. So thanks for this really cool setup. I think I'm going to ask the question that everyone has on their mind, and Victoria already sort of mentioned it. How do you think AI will help transform the field, or where do you think it can bring it to?
We decided that tough question I start to take while the other one think about the answer. So now you will get a good enough answer, then you will get the good one. No, I think AI has already started to transform the way we do science, because, for example, when we look at how an animals walk, right? When I started my postdoc, I used to do it manually. So you sit in front of a computer, exactly, and you just say, okay, this is a knee. And then the mouse moves a little bit, like this is still the knee. And then you do this, and in a day, you maybe manage to look at an animal that did 4 steps. Now we do, in the same amount of time, analyze 65 animals that have been running for 6 hours. Right. And you don't have to sit in front of the computer. You just sit for a bit, you train artificial intelligence to do what your eye can do. And of course, it's not as perfect as your eye, but it's good enough so that you, that definitely has accelerated science, but also has made it more feasible to focus on the result rather than on the technical build-up. And I know that, for example, now AI has been used also for developing models that can make understanding the brain, or recognize pattern in a way that maybe our eyes will take much longer. So for me, definitely the speed, speeding up research has been essential for the AI part.
I have a very specific vision, I feel, how AI, will help. I think, when we think about animal research and can we create a virtual rodent model that lives in this, but then likewise, right?
Like a digital twin?
Like a digital twin, right? But then can that be interposed on whatever model or network we feel in the human, right? And so I think with the virtual rodent model, we're actually really close. There's people the International Brain Lab that are recording from all these different places in the brain, right? And creating these models of how the rodent brain works. And so it has I think it's an incredible tool, right? People can mine that data for ages. And so I think that's sort of my vision, where for example, we can take all the EMG data and create this virtual model that we can probe. It's an amazing hypothesis generating tool, right? And then you can yes, of course, you go back to your mouse and you test it. But I'm incredibly excited about that. Yeah.
My name is Richard, and it's a follow-up question on AI as well. Now, we talked about technology, and I'm interested in knowing, how do you think AI will help us understand motor intent to implement technologies like brain-computer interfaces?
This is maybe a little bit contrarian of me, but I actually think that the impacts of AI have been really on the quotidian, day-to-day, how much you can get done. I mean, this is really following up on your answer. So, what we think about when we think about the future of AI are these, moonshot things of, new ways of doing science. And I think I'm actually very excited about those. But, in my lab and in the lab of people I work with, the major impact is just you can get more done in a week. And you have to very carefully, I mean, I think there's an interesting training issue here. Like, it's a major, and I think this is maybe a little bit unacknowledged. I and my colleagues, we're teaching people in our lab how to use AI coding assistants in a way, it's great at generating something, but I think, the impacts now are really, in some ways, like I said, very prosaic and very immediate. But I think it's really incumbent on us to train, undergraduates and graduates and postdocs how to be responsible AI users, not in the sense of not destroying the world, although that's important also, but, to teach people how to, the ways in which AI does not replace intuition, does not replace creativity. But if you use it in the right context, keeping those sort of, really old school scientific virtues intact, you can just be much more effective and much faster. So it's not again, this is not sort of a poetic, the future is here today thing, but I think that's the reality. And I think it's also the challenge we have to train the next generation.
So to your question, how do you understand intent, right? And it comes to what I said before. AI is very good at being trained to recognize pattern. And if we see, okay, if the muscle is about to move in that direction, if my hand is already going this way, it will kind of finish the movement, then we can use this to train AI. But I agree, the training is not just on the training the AI, is also training the trainer to use it as an accelerator, a speed up, but not as a substitute for critical thinking. And so, we have to first understand this pattern, and then we can train somebody to use this pattern in the correct way.
Maybe can say something. Yeah, it's a great question. And I think it's for me also, I mean, brain-machine interfaces or brain-computer interfaces have been, the promise for restoring movement, or at least through prosthetic devices, by recording signals from the brain and transforming it into robots moving for us or assisting our body. And I think there is a lot that science can, or that translational science can accelerate there with AI techniques as well. And I think what I would like to see is for society to put pressure on tech companies, because there is, Meta that runs a company called Control Labs, and they have a technology that came out of basic research, and they bought up that records from the muscle of the arm with a wristband, and then they control mouse on your computer and the control devices, and they're going to use it, potentially to make money, or definitely to make money. They're going to use it to sell you the next, smartphone that you don't have to have, but you just go like this and things happen on your computer, and which will be great for patients if it can be used by a patient. But I think it will be on academic researchers and the public and universities and hospitals to try to also do that, and kind of convince the government that it should be regulated in a way that it becomes also something for the good of patients and humans, and not just shareholders.
Actually, this was perfect. Hi, my name is Alex McGold. I have a question. How do you see the role of IP? So it's a sticky subject, especially when it comes to grants, because some of them actually, like AIDS Foundation and others, require you to go fast with publication, open access, and then we all know what we sloppily call the "we own you" contracts that most of the institutions demand that we all sign, and so sometimes that's a little bit hard to navigate. Thank you.
So the question of, first of all, how do you, if you have a new technology, how do you share it? Do you keep it secret for a while? And, partly, part of this was because I had no idea what I was doing, but also it was an intentional thing is, the idea that you can record signals from the brain reaching the muscles. We did not make that up. That has been around for, a really long time, but, I had an intuition that there was a much better way to do it. But, the first devices did not work that well. And it was really by pushing through, this international collaboration that we were able to develop it really fast. That actually helped us in the IP process, because if we'd never collaborated, we would have been in this serious problem of like, okay, we need to publish the papers, but we don't, but the thing is not ready to patent yet. So, I mean, I think it's, and there are different challenges for medical devices versus drugs and things like that. But I think that faculty in general need help both domestically and internationally with negotiating this. So the patent on our thing has been it's a patent application, excuse me, but it's been nationalized in Canada and the European Union. And I'm only now learning the US IP laws and implications, but we actually have not half, but maybe 30-40% of our users are outside of the US. So I have my immediate concerns of starting a company and licensing the patent, but then we have all of this upcoming stuff that we're going to have to negotiate or deal with in terms of the United States has this very unique healthcare system, maybe but and it's very different in Germany and other parts of Europe. So I mean, I think what I'm contending with is it's a particular case of this very general thing, which is that when an academic creates something that has a medical application, they are generally completely unqualified, and in some cases not supported by their institution. Yes.
Okay. I have to advertise how great my institution is at that part. No, because I think this is very important. As a scientist, what Sam said is that we are completely untrained to understand the concept of patent, IP, and so on. And now, for example, I'm receiving electrode from Sam. It's taking a while to write our agreement on IP because both our legal offices are involved. But then what we have in Cologne, which I think is great to support transfer and is also an educational value, it's called Gateway. And it's a structure that it helps transfer your idea to the market. So it's like an incubator, but also offers feedback and consultants. And so I know like one of our students was thinking to patent his idea, but then they offered him, okay, this is how the intellectual property works. This is how much will be your share, because of course your PI still has to be involved because they were mentoring you while you were developing your idea, which is, sometimes I don't, but these are the rules, right? So there is a lot of that we have to learn. And I think it's also important for the student when they approach the lab to start learning about this, because then they come in with the awareness on, okay, if I want to take this route, this is what I need to know, and this is my rights, and this is the best way to do it.
I have to advertise for NYU. Also, we actually just received a ranking of number 8 in the country for Ignition Schools, which is from Fast Company, I think. And that really is about how much we foster innovation with students, staff, and faculty. We have this whole entrepreneurial institute to help people start up startups, and really a really successful portfolio of getting, IP out into the world. So we can collaborate on that, maybe with University of Cologne. At this point, I think it's time for us to move to the reception, so we can continue the conversation. If you still have questions, we will all still be around. So I want to thank our panelists. This is really great, and thank you for this wonderful idea and bringing it together. I want to thank our organizers, the University of Cologne, NYU Langone, the German Center for Research and Innovation, and others. And I want to thank all of you for being here and taking part in tonight, and look forward to more conversation with a little bit of wine, beer, and food right outside those doors.